So I'm happy to discuss the point of departure working group. Myself from Corteva Agriscience, Jason O'Brien from Environment and Climate Change Canada, and Scott Auerbach from the NIHS will be leading this discussion. The general agenda here is I'm going to give an introduction to the project, what's our vision, and then what we accomplished in 2023. Jason will then go through what we're currently doing from a methods manuscript perspective, and then Scott will bring us home from our next steps and also discussing some ancillary projects. So next slide, Connie. So this group started in 2019, and we developed first a problem statement. So what are we going to do? And this is our problem statement that we developed. So we want to make a framework to derive an in vivo transcriptome POD. So what are the methods that would be used to do this? What are the optimal methods to do it? And really, this POD would then ideally be used in chemical risk assessment. So risk assessments that are currently based upon hazards, but are really based upon the most sensitive hazards. So can we use a molecular POD in lieu of that? And these PODs would then produce a health protective POD based upon concerted molecular change. So that POD is really based upon a constellation of molecular changes, not just one or two that happen to be the most sensitive, but a constellation of concerted change. So that's what we've been striving to do for the last 4 years or so. Next slide, please. And we've had a lot of participation. This has been a very satisfying project to lead over the last 4 years. People are highly engaged. And so we have cross-sector participants in industry, academia, nonprofits, as well as government. And we've had very productive discussions throughout the 4 years. So thank you all very much for your participation, and really happy to have others join our group as well. Next slide, please. So what did we achieve in 2023? So we had a roundtable session at the SOT. Carol Yock, who's a participant, she was a chair along with Kim Buckelhide. Myself and Scott were speakers, as well as other people outside of our group. It was really to discuss, what's the status of this transcriptome POD? I mean, are we ready to use it in risk assessment? And there was a large audience, a heavily engaged audience. At the end of that, Kim Buckelhide actually had a straw poll asking people, so is this ready for use, or should it be used at some either now or in the future? And I think about 90% of the participants in the audience raised their hand affirming that. So it was really, really nice to see. This project, although we have core functions that spurred related work, I'm not going to go into this because we have additional slides on these later on. And then an additional slide we're going to go through as well, but so I'll just touch upon it here. Connie and Leah Weymouth are co-chairing a workshop at SOT this year, or upcoming 2024, on the application of molecular points of departure for risk assessment across multiple molecular data sets and data types. Next slide. So I wanted to highlight, give some more background on what our goals are here, and our initial couple of years' work was outlined in this vision article that was published in Toxicology last year. It was again a multi-author effort. I really want to thank everybody for it, and it outlined our vision for how these omics data can be used in chemical regulatory assessment. The paper is open access, so please take a look at it if you'd like. Next slide, please. So in that paper, we outlined our vision, and it was really based upon 4 principles, which I'll briefly go through. But it's also based upon, I think, sound science and AOPs. So, a generic AOP is you first have exposure that, given a sufficient exposure, produces some molecular change. And then those molecular changes can induce an apical effect. And currently, the risk assessments for chemicals are based upon those apical effects. Our idea is that we can take a step back and use the molecular change point of departure for risk assessment. So the principles that we outlined are transcriptomics is a reliable tool for altering or for examining alterations in gene expression. I think that's a sound principle. supported by a lot of data. Principle 2 is that these alterations in gene expression are indicators of these apical effects, whether they're adaptive or adverse. I think there's also very good science supporting that principle as well. Principle 3 is really where the project is currently focused, and that is the principle is that transcriptomics can be used to employ to establish a benchmark dose-based point of departure from short-term in vivo studies. And that this point of departure is based upon concerted molecular change. So there's no concerted molecular change below this point of departure, and therefore no apical effects. So we're really trying to identify methods that can establish that principle. And then the last principle is that this transcriptome POD will provide strong protection for human health and really environmental health as well for use in risk assessment. So I will turn it over now to Jason. He's going to go through our current work in 2023 around a manuscript on bioinformatics processes. Go ahead, Jason.
Thanks, Ken. While we were working on that first manuscript that Ken just described, we had a subsection describing the data analysis workflow of the general approach to establishing a molecular point of departure. And while we were working on it, we realized that there was a lot to be discussed about this pipeline. Now, there's the overall workflow has generally been established, and can be it's shown in this little diagram here. I won't go through it in detail, but these are some very common steps along a bioinformatic analysis for establishing a point of departure. And while these steps are common amongst most studies, the particular details of each step, there's a lot of variability in the literature to how these are done. And so it became clear to us that in order for this to be used for regulatory purposes, there needs to be some kind of, if not standardization, then at least some guidance documents on how to address all of the different options and make decisions for a reliable point of departure. And so the current manuscript is to take a look specifically at this bioinformatics workflow, look at all of the different options along the way, try to establish, identify some of the data gaps and things that we just don't completely understand throughout it, and make some recommendations. So right now the manuscript is in a pretty close to finished state. I believe it's going to be going for internal reviews at various organizations. And then we'll submit it for publication, of course. But then the end goal is to take what we've learned from this manuscript and get some people, experts in the area, together for a workshop to specifically address some of the challenges that we've identified. And this will involve a few steps. First, we need to identify and make a collection of data sets that are appropriate for addressing all the different kinds of questions. And then we will assemble, I think we're going to do like a working type workshop where we have practical problems that need to be addressed. And these will result in a series of manuscripts addressing all of these different challenges. And there's a wide variety of challenges. Most of the bigger challenges are related to the sensitivity and the specificity of the method. There's kind of a gray area in between what's a negative response and a positive response, what's a weak response, and that requires a lot of thought. There's also some challenges with describing the confidence, empirically, of the method, as well as taking a closer look at the mechanistic interpretations. Although generally there's kind of two types of points of departure, a mechanism-based one and a mechanism-agnostic one. And largely this first paper has argued that the mechanism-agnostic one is probably going to be protective in most situations. There seems to be a strong desire to understand the mechanism and try to get more mechanistic information out of this. And we've kind of determined that the methods that we are using might not be the best way to pull out and tease out specific mechanism information, and so there's some work required there. And so this workshop will kind of address these issues and hopefully come to some concrete conclusions and recommendations that we can make going forward. That's about all I have to
Great.
say about that.
Great. Thanks, Jason. So a couple of slides on ancillary projects. Jason mentioned, what's the sensitivity and specificity. So there's a project that Corteva and Syngenta and Siom have been doing to try and tackle that question on some level. So we've been working on a project, a multi-organ rat multi-organ transcriptome project with the purpose of optimizing bioinformatics processes to drive a TPOD. And this is taking a large number of 22 animals, just control animals, unexposed, putting those data through the TPOD process and seeing, when do we generate a point of departure? What kind of processes can we tweak to minimize the false positives while still maintaining appropriate sensitivity? So that the project is examining the organs shown in the tables here, and we've been doing the analysis in a few different ways, both from fresh as well as FFPE samples and using TempoSeq and RNA-seq. This project is nearing completion. The manuscript is currently being written, and there'll be more to come from this. But this is one project that's been spurred by the POD group. So I'm going to turn over to Scott now to take us the rest of the way through the project.
You're muted, Scott.
There we go. Next slide. All right, so we just wanted to highlight some of the work that's being done by members of the group that speak to the practical aspects of things. And this is something put forward by members of Syngenta that are in the POD work group. And it's a workflow on how they anticipate integrating TPOD-based assessments into their assessment pipeline. So you start at the top, you have a new chemical. Assuming it's non-genotoxic, you actually travel down this path. If it's genotoxic, you move directly down and evaluate a weight of evidence and conduct additional health assessments. But assuming you're not in a non-genotoxic, you have a non-genotoxic chemical, you first evaluate whether or not there's a human relevant species or not. If there isn't a human relevant species, you conduct short-term studies to evaluate TK and TD. If there is a human relevant species, you pick that species. The example put forth by them is an HPPD inhibitor in mouse, as mouse is the relevant model there. And you again do the TK and toxicodynamics. After which you evaluate, you conduct a weight of evidence for the molecule to inform the subsequent study design. And this, of course, not surprisingly, would look different if it was a new versus an established chemical. At that point, they would conduct their studies at a steady state level. So they basically evaluate their histopathology and their transcriptomics once they've reached steady state dosing levels, and they would evaluate the targeted organs that were identified by the weight of evidence analysis above. And then their assessment of transcriptomics will either be from FFP or fresh, and I'll touch on that next. From there, they derive BMDLs for the apical effect target organs and the transcriptomics, and conduct a weight of evidence assessment based upon the combined genotype, phenotype, and the potencies. And then either the weight of evidence supports sufficient data, indicates sufficient data has been generated to conduct a health protective risk assessment, or alternatively, there needs to be more work done. So this was a really nice piece of work that they put together that helps us sort of understand how they're thinking about the use of TPODs. So next slide. Another just feasibility issue when you're thinking about how you integrate the TPOD approach into your testing pipeline, in particular the integration with guideline studies. There's issues about sampling histopathology and then also being able to evaluate transcriptomics at the same time. So they looked across a collection of tissues they typically evaluate in their shorter term in vivo studies. And they identified whether or not it's possible to get fresh tissue or FFPE from those tissues. And they noted when they couldn't get fresh tissue, they noted why. So this was just, again, just important thinking through the implementation of this process. And what they did find, and what's great, and Leah's work I think speaks to a lot of this, is they have they do believe that they can pull samples from FFPE for all the critical tissues. Next. So we already heard about this, but I just wanted to go a little bit more depth. The group of folks in the POD group put together a workshop for SOT called Molecular Points of Departure, Transcriptomics and Beyond. The goal is to discuss the use of multi-omic and alternative measures for identifying POD values and the added value of integrating the response for better, integrating the information for better risk protection. The chair of the workshop is Leah Weymouth from US EPA, and the co-chair is Connie, and then you'll all recognize the speakers. And I really want to congratulate Leah and Connie for, I don't see myself, Rusty Thomas, or Carol Yawk as speakers. So I think that was we found some new blood to discuss this issue. So congrats, thank you. Next. So this, I wanted to highlight this because it really is, it's very much a byproduct of the efforts of a lot of the people in the POD group. So this is an effort led by CCTE at EPA to push forward a new health assessment product for data port chemicals. So you see on the left there, everyone's familiar with IRIS and PPRTVs and other assessments. And EPA saw an opportunity for, actually collectively putting together the efforts of many people in this group and formulating a product called an ETAP, or what they call an EPA transcriptional assessment product. So they've gone through the process of formulating a number of documents describing what they're planning on doing, and they've held Board of Scientific Counselors meetings. Specifically, there will be there was an ad hoc board review that I was a part of to evaluate the actual ETAP product itself in July. And then subsequently, they did a pretty deep dive in a value of information assessment comparing ETAP, which basically is a 5-day toxicogenomic study, very similar to what a lot of people in this group have been doing, relative to the cancer bioassay. And it was, a lot of it was weighted on the amount of time that was required to get an answer on a point of departure that is health protective. Next. So this just lays out the product formulation, or the ETAP product formulation, the goal of which, again, is to develop and operationalize a new US EPA human health assessment product for data port chemicals that can be completed from chemical procurement to publication in less than 9 months. So the less than 9 months is really what is outstanding in this case, and really drives the value of information. So the first 6 objectives here are related to the ETAP itself, and the 7th is, of course, the VOI. So objective 1 is review the relevant literature, which, again, many people in this group have contributed tremendously to this literature, this corpus of literature, and it was heavily cited in the document. Then they wanted to refine the dose response analysis methods for a standardized study design. Again, the critical thing here is they wanted to get a very detailed study design written down and peer reviewed. So there's basically, the only thing you really have to do is generate the data, run it through the pipeline, and get it QA'd and then published. So there was a specific goal of avoiding excessive peer review. So again, they've developed a very formalized process for analyzing the data. They also went through and compared error and concordance with variability in the toxicity studies. So, how variable is your point of departure, whether you do a 5-day transcriptomics versus, say, a 90-day or a 2-year. Develop standardized methods. Again, that's the standardized methods are just a formalization of how they're analyzing the data. So it itself can go through the process, can go through peer review. The analysis process can go through peer review, but and so the subsequent products that come out evaluating individual chemicals, they're already pre-reviewed for how they're analyzing the data. And then they went through and compared, they took the pipeline that they developed, they compared the transcriptomic reference values with the traditional RFDs, and then developed an example ETAP that the reviewers could evaluate. And it was done on a data port PFAS. And then finally, they conducted a very elegant value of information assessment, looking at the socioeconomic case study on human health and economic value of the ETAP relative to the 2-year cancer bioassay. Next. So this just summarizes the the ETAP product and how it's structured. And it's very similar to what we did at DTT a couple of years ago, where we pre-reviewed the analysis approach. And then we had a standardized template for how we developed a standardized template for how we're reporting. And so this standard methods document is obviously more specific than a normal guidance. It has very delineated parameters for how they're analyzing the data. That methods document is subject to peer review and public comment. And again, the whole point of this was to focus on data poor chemicals. The other component is, again, the highly standardized template that goes through, has minimal free-form text, no subjective interpretation. So there's no pathway analysis necessarily. There's no linkage to pathological manifestations. It's purely a potency assessment based upon the analysis pipeline. It's then reviewed for quality and consistency with methods by the EPA QA staff, and then there's internal technical review by the ORD scientists. And again, the really powerful thing about this, there's rapid experimental execution, streamlined review process, target time for initiation to release is less than 9 months, and is scalable, and it has potential broad application. Considering what we've seen so far in the generalizability of the approach. And Connie, I was making a joke, by the way. I just saw your email. I was making a joke. I'm actually really glad you didn't include us. I'm glad that other people are picking up the sword and running forward. This is great. And then, so from 2024 to 2025, we plan to finish and submit the paper that you heard Jason talk about on the bioinformatic workflow, and then plan a workshop to, as Jason talked about, to further delineate and derive answers to the many issues that we are facing and how we analyze the data. Next. So we'll point out there are some potential next steps and opportunities for anyone who's interested in joining and contributing. First of all, the opportunities. Well, we plan on sharing curated existing data, cancer data, with the eSTAR Carcinogenomics group. Jessica Ewald, and I don't know if Jessica's here. Jessica was spearheading the development of a public repository to deposit chemical POD data and data sets, so people could analyze them with whatever pipeline that they formulate. And then there is the scientific advocacy to facilitate adoption into regulatory decision making. And again, there's real leverage here with the EPA ETAP product as it's going forward, and if it gains full acceptance in the context of use that EPA is planning. And there's also also potentially now that, again, as we're making our inroads into the regulatory process, to collaborate with OECD and other stakeholders to socialize the approach and drive regulatory adoption. There are still some uncertainties, and Jason hinted at some of this in the bioinformatics component, but there are also questions on a grander toxicological scale. The degree of, one of the questions is how broad a tissue coverage do we need to have, what the exposure durations need to be. I know there's significant questions about developmental toxicity because the protocol is basically 6 to 8 week old animals for 5 days. So there's no developmental exposure. So the question is, are we sensitive enough to protect from developmental effects? And then the question is, do we need additional species? And then finally, what you saw earlier from Syngenta is their own thought process on how to integrate it internally into how they, the TPOD, and how they assess chemicals. But there's obviously the outward formalization that would be wonderful, assuming that it gains, this approach gains acceptance. How would you integrate it into existing testing paradigms? And is it possible to do actually create a fully independent assessment, say what the what EPA has proposed as the ETAP, or alternatively, how do we move it to an in vitro context? Next. Keep trying to move the slides myself. And we'll take any questions. Thank you.
Great. Thank you so much, the three of you. There were some conversations in the chat, and I know you answered it, Cam, but maybe it'd be worth just talking to the whole audience about what the CMC is and how we defined it, if you don't mind.
Yeah, I'm not sure we would find it well. It's a lot like, an orchestra warming up. You have, the French horn and the tuba player doing it, and then all of a sudden the orchestra starts playing. That's what it is. You know it when you see it. How you actually define it, I think we've defined it, and Scott and others can provide our perspective. I think we've defined it as, the dose where you begin, where the inflection point of molecular change increases greatly. So the slope of that change increases greatly. It could be defined from a mechanistic perspective as well if we knew enough about mechanism and thresholds in mechanism. But it's certainly not one molecule changing on its own, and it's probably a few, at least 100 molecules from a transcriptome standpoint changing. But
I think
defining it's a challenge.
Yeah, I think qualitatively we have a generally good understanding of what that means, but more specifically and quantitatively, I think that's something we hope to investigate in our workshop in defining the sensitivity of the method and specificity of the method. This is an important, outstanding question. Yeah, so we hope to take a closer look at that and maybe even provide a more concrete definition once we get some more quantitative definitions of this.
Now, there's mathematical and statistical means, and then there's the intuitive biological means of doing it. And I think that's our inclination is as biologists to do it biologically. But if you look at the way we've evaluated the data, looking at the concordance of the points of departure, it's been, and this is what EPA's put forth is a data-driven approach where you say, look, if I model the data this way, I get agreement with the 2-year point of departure. What that means biologically is going to be a challenge to deconvolute in some cases, but there are it's something that we need to set our goals on, and it's something that we're working on. And again, a lot of it is defining what the biological meaning behind change in certain gene sets or individual genes means. And so it's and we actually have a project underway that we're collaborating with this group here, at least the early stages, that was initially started at DTT to start curating mechanistic information associated with toxicogenomic responses from individual genes. Starting off with the easy one, KIM1, and a couple others that we know a lot about, but then trying to move into other areas to try to better understand what the biology is of those genes that are responding.
Great, thanks for that answer. I think we have time for maybe one more, and then maybe we could do the rest in the chat. And I know, Cam, you already started to do that, and others. So Vicky asked, if testing of agrochemicals is done after steady state has been reached, how would the potential short-term, early transient responses be taken into account?
So I think my perspective on this is that you need to reach steady state so that you.
Do we lose Cam? Is that just for me, or is that for everyone? We lost Cam.
Am I out too? No,
he's gone.
Yeah, I hear Scott and Jason. One of you want to step up and maybe answer.
Jason, you want to field this?
I don't know that I have a good answer. It's a great question that comes up all the time. I'm not sure if it's completely related, but a very common question that we get is how can you differentiate between adaptive and adverse? I'm not sure if that's touching on the same thing in your question. And I think part of the answer is that if regardless of whether it's if it's a adverse response or not, we can kind of see it as a threshold beyond which we don't want to pass before adverse effects. So in a sense, it doesn't matter if it's if it's what kind of response it is, but it's a point of departure beyond which adverse effects are expected. I don't know if that specifically addresses, because you're talking about more of before steady state, which is a little bit different, I think. And I think I would need a little bit more thought to address your specific question, but maybe it's related to the answer that I gave.
Thanks, Jason. Thanks for doing that on the fly too, as we lost Cam. So,
So just a question.
Yeah, go ahead.
Just a question for the individual that posed this.
It was Vicky.
Vicky? Oh, sorry. Yes. So we've done temporal analysis, looking at transcriptomic responses, and there are some, PPAR alpha activators are classic for this. Like there's a 24-hour jump, and then they drop down, but there's temporally covariant pathways that then are subsequently activated over time at about the same dose level. So you'll have the initial, I think it's proteasome, ribosome, and there's a little bit of cell cycle effect, and then it tapers off. But then there's other things that happen at about that same dose level. So I think in some ways they're just proxies to what's happening. Now you're dealing with a steady state issue, and that's an important issue. If you're not at steady state, now most of the chemicals are not that I think we're dealing with this approach, at least from currently from an agrochemical standpoint, they're not bioaccumulative. Obviously, we have issues related to PFAS and some of the chlorinated and brominated chemicals that are a challenge. But so just thinking through that transient response that may change as you go on is still it just leads to a secondary response and a tertiary response that are typically about the same dose level. So your point is your POD answer would be about the same in most cases.
All right. Well, we got Cameron back, but we are sort of at time. Any last comments from you, Cameron?
No, thanks.
Great. Thanks for that excellent conversation. I know we had a few other points in the chat. Maybe if Cam, Scott, and Jason wanted to take a look at those and see if there's ideas.